Alright, so this is Dr. Piri again. So today we'll be starting to discuss endocrinology. So I welcome everyone to endocrinology series of lectures that we're going to have. So this is quite a huge topic. It will take a bit of time for us to discuss all the endocrinology. So here we're going to discuss a lot of hormones, the glands that are producing the hormones, and also disorders that are as a result of overproduction or underproduction of hormones. So let's start with our information. So this is endocrinology or endocrine system. This is where we're going to discuss hormones like I've already said. So for starters, we're going to start with introduction, then we'll look at organization of the endocrine system, then the nature of hormones and mechanisms. So what are the nature of hormones? So the general nature of hormones and the mechanisms that are involved. So each hormone, it will have a particular mechanism which is going to stimulate other cells to respond. So what are the mode of actions for these hormones? How do they communicate with other cells? How do they have an effect on other cells? Via the receptors, of course. And towards the end of our endocrinology classes, we are going to discuss the endocrine disorders. So if there is overproduction of a particular hormone, there is underproduction of a particular hormone for some reasons, what could go wrong and what are the disorders or the pathology that are associated with these hormones. So that's basically everything that we're going to discuss in endocrinology. But you need to take note that the first part of endocrinology, we are just going to discuss the general endocrine system. So the general hormones and the glandular cells that are producing these hormones in the glands. Then towards the end, we are going to discuss reproductive physiology, where we're going to discuss more of reproductive hormones. So there are those hormones that are produced by the testes, by the ovaries or the gonads. So those hormones that are produced by the gonads, they are called reproductive hormones. So that we'll discuss under reproductive physiology. It's quite interesting, especially the female reproductive system. Okay. So now, the general principles of endocrine system. We all know that the major function of the endocrine system is to maintain the whole body homeostasis. So the whole body homeostasis is maintained by the endocrine system. So there are two systems which are the control systems of the body, the endocrine system and the nervous system. So the two systems are the ones that will control the function of the body. So they're going to control the function of the body via the coordinated or the coordination of hormonal signaling pathways that will regulate cellular activity in target organs throughout the body. So throughout the body, there are target organs. The target organs, they will contain target cells, and those target cells, they are supposed to have specific receptors for the hormones we're going to discuss. So for a cell to respond to the hormone, it needs to have a specific receptor to receive or to interact with the hormone. So the hormone will go and bind to the receptor, then it will trigger some cascade of reactions within the cell, then the cell will be able to respond. So the two control systems of the body are the endocrine system and the nervous system. So the multiple activity of cells, tissues, and organs of the body are coordinated by the interplay of several types of chemical messenger systems. So there are chemical messenger systems like the neurotransmitters, the hormones, the first chemical messengers, the second chemical messengers that are found within the cells. So all those are going to control the activities of the cells, the tissues, and the organs, and the body at large. So they are controlled by these chemical messengers. So what are these chemical messengers? So some of these chemical messengers could function as neurotransmitters that are being produced by nerve endings or axon. So you have nerve endings that can release neurotransmitters. So some of these neurotransmitters, they could be excitatory or inhibitory. We all know to say that excitatory neurotransmitters, they are going to stimulate the production of excitatory postsynaptic potentials. The inhibitory ones, they'll bring about inhibitory postsynaptic potentials. So you find that if the nerve cell is affected by the inhibitory neurotransmitter, it's going to be inhibited to fire an action potential. But if a nerve cell is stimulated by stimulatory neurotransmitters, all those are going to stimulate the cells so that they can fire an action potential. So you find that the postsynaptic nerve cell is going to fire an action potential, and these are signals that are transmitted from one point to another point along the axon of the nerve cells. So some of these chemical messengers are neurotransmitters. So neurotransmitters are released by axon terminals of neurons into the synaptic junctions, and they act locally to control nerve cell function. So these are chemical messengers or neurotransmitters that are released by the axon terminals. So once they are released by axon terminals, they will go and attach to receptors on the postsynaptic membrane. So on the postsynaptic membrane, it can either bring about inhibitory effect or stimulatory effect. So if it's an stimulatory neurotransmitter, you'll find that it's going to stimulate the postsynaptic nerve fiber. If it's inhibitory, it's going to inhibit. So you know to say there are different types of neurotransmitters. There are those that are going to stimulate, like acetylcholine. You have those that are inhibitory, like glycine. It could be inhibitory, and gamma aminobutyric acid. Gamma aminobutyric acid could be inhibitory. So those that are inhibitory, they can cause influx of negatively charged ions like chloride into those nerve fibers. So if those nerve fibers are gaining negatively charged ions, it means that the membrane potential is becoming more hyperpolarized. So it's very difficult for these cells to be stimulated to fire an action potential, because the membrane potential is moving away from the threshold potential to which this nerve fiber is supposed to fire an action potential. So those are inhibitory. So neurotransmitters are also classified as chemical messengers. Then we also have the endocrine hormones. So these are the things that we are going to be discussing in this system, the endocrine system. So we have the endocrine hormones, which are released by glands or specialized cells into the circulating blood, and they will influence the function of cells at another location in the body. So they need to be released into the circulation. They will be transported by the circulation to a target cell, a distant target cell that will contain the receptor. So these are the endocrine hormones. So they will have an effect or an influence on other cells. So those cells will be able to respond. So the endocrine hormones are also chemical messengers. And of course, we have neuroendocrine hormones. The neuroendocrine hormones are secreted by neurons into the circulating blood, and they will influence the function of cells at another location. So at another location within the body. So they will have an effect as well. Okay, so the neuroendocrine hormones are secreted by neurons into the circulating blood and influence the function of cells at another location in the body. So they need to be transported by blood. Okay. Then we also have paracrine. Paracrine are secreted by cells into the extracellular fluid and affect neighboring cells of a different type. So paracrine, we saw a lot of paracrine communication when we were looking at GIT, the gastrointestinal tract system, whereby there were some cells that are producing certain chemical messengers, and those chemicals will have an effect on neighboring cells of different type. So you saw that we had that cell that was producing a lot of histamine, and histamine had an effect on the parietal cells that were producing hydrochloric acid. Then we also had the G cells that were producing gastrin, and the same gastrin had an effect on the parietal cells and other cells that were producing certain products in the gastric pits. So those are paracrine. So para means nearby. So you have a cell that is producing a chemical messenger that will have an effect on the neighboring cells. So this is paracrine. So they are also chemical messengers. And then autocrine. Autocrine is whereby a cell is secreting substance, and that substance will have an effect on that particular cell producing it. So it's called autocrine. So autocrines are secreted by cells into the extracellular fluid and affect the function of the same cells that produced them by binding to cell surface receptors. So the same cell that is producing a chemical messenger, it will have an effect on that particular cell producing it. So it's called autocrine. So it will have an effect. An example that I can give here are the tumor cells. you realize that there are certain tumor cells that are always growing, invading the body cells. So how do they manage to do that? You find that those tumor cells or cancer cells, they are producing chemical messengers that will have an effect on those particular cells for them to continue growing. So that kind of communication is called autocrine. So they are using growth factors as chemical messengers to stimulate the growth of this particular cell producing it. Cytokines are peptides secreted by cells into the extracellular fluid. So you have cytokines that are produced by different types of cells, especially immune cells. So immune cells, most of the immune cells could be producing a lot of these cytokines. So cytokines are peptides secreted by cells into the extracellular fluid and can function as autocrine. So they can function as autocrine, they can function as paracrine or endocrine hormones. So examples of cytokines will include interleukins. So you saw when you're looking at erythropoiesis or in general hematopoiesis, where the white blood cells were being produced and the red blood cells. So there were a lot of cells that were producing cytokines that will have an effect on those cells that were developing within the red bone marrow. So some of them are interleukins that are being produced there. So we have interleukins and also other lymphokines, so that are secreted by helper cells and act on other cells of immune system. So the cytokine hormones, an example is leptin hormone, of which we also discussed when you're looking at the gastrointestinal system. Leptin hormone is being produced by the adipocytes or adipose tissue. So the adipocytes, they are adipose cells that are producing leptin, and the leptin has got an effect on the hunger center and satiety center. So they are involved in food intake regulation. So you know to say that leptin is going to inhibit the hunger center so that you have satiation taking place there. You feel satisfied because you have a lot of leptin that is producing and that is being produced by the adipose tissue. So sometimes they are also called adipokines. So the leptin sometimes is referred to as adipokines because they are cytokines produced by adipose. So intercellular communication, how do these cells communicate? One cell communicating with another cell, that is intercellular communication. So cells do communicate with one another through different channels of communication. So there are direct channels of communication whereby one cell can communicate with another cell. An example that I can give here are the interleukins, or you also have selectins, you have cell adhesion molecules. So the cell adhesion molecules can also be used as means by which cells are going to communicate. So they are direct channel of communications. We have gap junctions. One cell is able to communicate with another cell via a gap junction. So cells do communicate via direct channel of communication, and sometimes we do have specific contact between cells, like the cell adhesion molecules, like I've already explained, to say that we have specific contacts by which the cells are going to communicate with one another. Then intercellular chemical messengers. The intercellular chemical messengers come in here due to the function of hormones and neurotransmitters or any other chemical messengers. So the chemical messengers are released into the intercellular spaces or the interstitial spaces, then they are able to have an effect on other cells. So these cells are able to communicate using the chemical messengers. So these are different ways by which cells are going to communicate, of which I've already explained that we can have autocrine, whereby a cell is communicating with itself. So it's producing certain chemical messengers that will have an effect on that cell producing it. So here you can see that you have this cell that is producing a chemical messenger that will have an effect on that cell. So this cell producing it, it will have a receptor. So these receptors can interact with a chemical messenger. So this cell is going to respond by growing or whatever it is. So there is cellular response, autocrine. Paracrine communication, you have a cell that is producing a chemical messenger, and these chemical messengers will have an effect on nearby cells of different type. So you can see you have these nearby cells of the different type. They are expressing the receptors for this chemical messenger. So it's coming from nearby cells. So this is called paracrine communication. Then we have endocrine communication. This is the major topic for this particular endocrine system. So we have the endocrine communication, whereby the glandular cell is producing a chemical messenger in form of a hormone that needs to be transported by the circulating blood. So via the circulation, it will be transported using the bloodstream from the point of production to a point where you have a target cell. The target cell is supposed to have a specific receptor for that hormone for it to respond. So this is called endocrine. Endo means within, crine means to secrete. So endocrine, it means the chemical messenger is being secreted within the body, within the internal environment of the body, hence the name endocrine, as opposed to exocrine. Exocrine, you find that what is being produced to exit the body, so it will be released to the outside of the body, be it in the lumen of the GIT or on the skin. So those are exocrine. They do have a gland system, they have a duct system, but the endocrine system, they don't have a duct system. So they are not transported by duct system, but the chemical messengers released into the interstitium, then transported by the circulating blood. So it's basically the same information even here. So you can see that we have the gap junctions. So two cells are communicating via gap junctions. So you have direct communication from one cell to another cell. The response that you're going to get there, it will be just a local response because just two cells communicating. So you have a local response. Then in terms of the specificity, dependent on the anatomical location. Okay, so you can have a communication between two cells. So for instance, smooth muscle cells, they do have gap junctions. So the smooth muscle cells that are lining the lumen of the GIT, they do have a lot of gap junctions, so they are able to communicate. So if one cell is stimulated, it can also stimulate the next cell. So they are going to function as a single cell. That's the syncytium functioning of cells, because they do communicate via gap junctions. But the response that you're going to get there, it's a local response. It's not a general response. Synaptic transmission. So you can see here synaptic transmission, you have the axon terminal that is releasing neurotransmitters that could be stimulatory to stimulate the postsynaptic nerve fiber via the receptors. So the two cells are communicating, the response will be local as well, but the transmission of the message has to cross the synaptic cleft, this space between the two cells. Then you have paracrine and autocrine, of which I've already explained. To say autocrine, one cell communicating with itself, so it's producing chemical messengers that will have an effect on that cell producing it. Or you have paracrine, whereby a cell is producing chemical messengers that will have an effect on nearby or neighboring cells of different type. Endocrine, a cell is producing a hormone, a chemical messenger that needs to be transported by the circulating body fluids like bloodstream. Then the response that you're going to get there is a general response. Why? It's because all those cells, the body that contain the receptor for that particular hormone, they're going to respond. So you're not going to get a local response, but to be more general, because most of the cells are going to respond as long as they are expressing those receptors on their plasma membrane or inside of the cells. So we have different locations of the receptors. We have receptors that are located on the plasma membrane of the cells. We have also other receptors that are found inside the cytoplasm of the cells, and other receptors that are found within the nucleus of the cells. So all those receptors are going to respond depending on the hormone that has been released here. So it's dependent on the receptors. They are specific for the receptors. Okay, so in this lecture, we're interested in the endocrine. So the paracrine, autocrine, this is where we end that information, and then we'll be just biased towards the endocrine system. So the endocrine system, like I said, the endocrine, the word itself, endocrine, endo means within, crine means to secrete. So secretions within the body, endocrine system. So the endocrine system is a collection of glands of an organism that secrete hormones directly into the circulatory system to be carried towards distant target organs. So the distant target organs are the ones that will contain receptors. So once the endocrine system or glands have produced a hormone, this hormone will have an effect on the target cells that are expressing the receptors, like I've already explained. So the major control systems in the body are two. So we have the nervous system and the endocrine system. So the nervous system and the endocrine system are the two control systems of the body. So they're the ones that are going to be involved to maintain homeostasis. So maintenance of constant internal environment is mainly done by the endocrine system and the nervous system. And the two systems, they are going to shake hands at the level of the pituitary gland. So at the level of the pituitary gland in the brain there, that's where the endocrine system and the nervous system, they are going to shake hands. So we'll see how they are going to interact at the level of the pituitary gland later on as we discuss lectures in endocrine system. So the nervous system uses electrical and also chemical transmission of signals. So the nervous system can use electrical in form of action potentials. We know to say that there is propagation of action potentials along the axon of nerve cells. So they are going to use electrical signals to communicate. The nervous system can also use chemical transmission. So chemical transmission of signals, we are talking about neurotransmitters. But the endocrine system, because it's mainly using the hormones, so it's biased towards the chemical transmission. So the endocrine system uses chemical transmission of signals to communicate with the target cells. So they are using the chemicals, which are hormones in nature. So these hormones could be proteins, or they could be derivative of amino acids like tyrosine and also tryptophan. So some of these hormones could be steroids, they could be proteins, they could be derivative of amines. So these are chemicals that will have an effect on target cells. The endocrine glands are also called ductless glands to distinguish them from all other exocrine glands. So the exocrine glands, they do have a duct system. So whatever is being produced by the acinar cells, they will be released into the duct system for transportation of the chemicals. Then the release of these chemicals is outside the body. It could be in the lumen or the GIT, which is not considered as the internal part of the body, or on the skin. So you're talking about sweat glands, tears, saliva production, pancreatic secretions. All those could be exocrine as long as they have a duct system to transport what is being produced. So the ductless glands are called the endocrine glands. So they don't contain a duct system. So whatever is being produced by the glandular cells, it will be released into the interstitium or interstitial fluid. Then they can penetrate the blood vessels because the capillaries, they do have a lot of openings, the capillary pores or openings that can allow diffusion of these chemicals from the cells that are producing them into the cardiovascular system for transportation. This is a slide that is showing a comparison of endocrine and exocrine. So how can you compare the endocrine and exocrine? So most of this information I've already mentioned. So the endocrine glands, the secretions will enter the interstitial fluid and then they'll be transported by the bloodstream. But the exocrine gland, so endo means within, crine means secretion, to secrete. So secretion within the body. The exocrine glands, exo means outside, crine means secretion. So secretions of these chemicals outside the body to some extent. So whatever is being secreted here, the substances, they will need to enter the duct system and ultimately they will exit the body. Hence the word exo as opposed to endo, because the hormones, they will stay in the body. They will be released into the body and then they will stay in the body until there is metabolism and also excretion or clearance of these hormones. That can also involve the kidneys, they can involve the liver itself or the lungs for metabolism of these hormones after they have done their function. Examples of endocrine secretions or hormones, these are growth hormones, insulin, adrenaline, estrogens, testosterone. But example of exocrine secretions or chemicals, it could be mucus, saliva, and other digestive secretions like sweat, tears. So you have other digestive secretions that are coming into the lumen of the GIT, like pancreatic secretions, you have gastric pit secretions, you have intestinal secretions, you have bowel secretions into the lumen of the GIT. All those are examples of exocrine. Even sweat is an example of exocrine, and also tears, though all these are examples of exocrine. So this is how we are going to compare the endocrine as opposed to exocrine. Okay, so this slide is basically the same, giving you examples of endocrine. So endocrine, the thyroid gland can produce thyroid hormones. So you have the tetraiodothyronine and triiodothyronine. Those are the hormones that are being produced by the thyroid gland. Then they'll be secreted into the interstitium. They'll be transported by the bloodstream to the target cells. Then we have the exocrine on the other side. An example is the production of sweat. So we have the sweat glands as an example of exocrine gland. So it's producing sweat that will be released on the skin. So you can see we have the sweat gland here in the duct system that will transport the sweat to the skin. So it's what is being secreted is exiting the body, hence the word exocrine, as opposed to endocrine. So the general endocrine system, there are glands that we are going to discuss that are producing the hormones and the functions of those hormones. So here we have the major glands that are producing the hormones. The pineal gland, we have the hypothalamus, the pituitary gland, the thyroid gland, the parathyroids, we have the thymus, the adrenal glands, the pancreas, the ovary, and the testes. So these are the major glands that we are going to discuss in our lectures. So to start with, you can see here there are 10 major ones. There are also other minor glands that can also produce hormones. So we'll be discussing them as well. But if a question comes to say, can you list the major glands that are producing hormones? Then at least you can list the 10 glands. But if I say in a male, then you need just to give me 9. Otherwise, a male, they don't have ovaries. And females, they don't have testes. So if I say in a male, you just need to give me 9. If I give you females, then just need to give me 9. Okay? But otherwise, if you have forgotten all these 10, there is something that can remind you the number of the glands that are there, or the major glands that are there, to that quadruped. So to that quadruped, to remind you all the major glands that are producing hormones. So T is for the testes if you are a man or male. The O is for ovaries if you are female. Then the other T is for the thymus. The H is for the hypothalamus. The A is for the adrenal glands. The other T is for the thyroid gland. Then we have the 4P, the quadruped. So the P's are 4. The pineal gland, the pituitary gland, the parathyroids, and the pancreas. So this will summarize all the major glands that are found in a human body to that quadruped. This picture is also summarizing the major glands and the products that are produced by these glands. So the glands that are there, the major glands now, and the products that they are producing. So as we go on discussing lectures, because we have a series of lectures in endocrinology, so these lectures will just expanding what is being shown here. So what is being shown here is a summary of the endocrine system that we'll be discussing. So we have the major glands, starting with the hypothalamus. The hypothalamus, they can produce ADH, antidiuretic hormone, oxytocin, and regulatory hormones. So the hypothalamus can control the hormones that are coming from the pituitary gland. So they do have inhibitory hormones and stimulatory hormones that can stimulate other hormones to be released or other hormones to be inhibited from the anterior pituitary gland and also posterior. But the anterior is the one that is producing a lot of hormones, because that's where you find glandular cells that are producing a lot of hormones. So we have the hypothalamus, then the pituitary gland. The pituitary gland, which is called the master gland, is releasing a lot of hormones. So we have the adrenal hypothesis. So the adrenal hypothesis is also called the anterior lobe of the pituitary gland. So we have two lobes. We have the anterior lobe, that is on the anterior aspects of the body. So the anterior lobe is producing a lot of hormones. This is where you have the glandular cells. Later on, you appreciate that the anterior lobe was derived from the primitive oral cavity, and you have adrenal. Adrenal means gland. So adrenal, that's why you have adrenal hypothesis. So the adrenal hypothesis, we have a lot of glandular cells that were derived from the primitive oral cavity that will be producing a lot of hormones. So you have the production of adrenocorticotropin hormone, the thyroid stimulating hormone, growth hormone, prolactin hormone, follicle stimulating hormone, luteinizing hormone, we have melanocortin stimulating hormone, and other hormones that are being produced by the anterior lobe. So these are the major hormones that are being produced by the anterior lobe. Then we have the neural hypothesis. Neural hypothesis, it means that it was derived from the neurons, so the diencephalon. So the neural hypothesis, it was derived from the diencephalon, which are basically neurons of the brain itself. Hence the name neural hypothesis. What is being released here in the posterior lobe. So the neural hypothesis is also referred to as the posterior lobe or the pituitary gland. There are two major hormones that are being produced there, which are also a good example of neurohormones because they're being released by neurons or nerve endings. So these are oxytocin and ADH. So oxytocin and ADH is not being produced by the posterior lobe, but it's being released by the posterior lobe. The production of these two hormones is taking place in the hypothalamus. So there are two nuclei in the hypothalamus. The supraoptic nucleus and paraventricular nucleus, they are the ones that are responsible for the production of oxytocin and ADH. Then they'll be transported by axonal transport, stored within the nerve terminals of these neurons in the posterior lobe. Then once you have a signal to cause the release of oxytocin and ADH, then they'll be released. So there will be stimulatory signals that can result into the release of ADH or the release of oxytocin. Then we have thyroid gland. The thyroid gland, they are producing thyroxine, which is T4 or tetraiodothyronine. They are also producing triiodothyronine or T3. So triiodothyronine is called T3. Then there are also other cells, the parafollicular cells, that are producing calcitonin. So we have the parafollicular cells of the thyroid gland that can produce calcitonin. Calcitonin is involved in metabolism of calcium or regulation of calcium. So calcitonin will be released when there is an increase in calcium levels. Then if you have a decrease in calcium levels, there is another antagonistic hormone that will be produced by the parathyroid gland. So you have the parathyroid hormones. So those two hormones are antagonistic hormones. So they will have an opposite function. So you appreciate more as we discuss lectures. The thymus undergoes atrophy during childhood. So you know to say in an adult you don't have a functional thymus, but during childhood you find that we have the thymus that is undergoing atrophy, but they will produce the thymoxines, and the thymoxines are more like hormones that will have an effect on other cells, as we'll be discussing. This is just a summary. Then we have the adrenal glands. The adrenal glands, sometimes they are also referred to as suprarenal glands. So they are called suprarenal glands. Why? It's because they are on top of the kidneys. The kidneys are called the renal. So kidney, it's a renal. So a gland that is on top of the kidney is called suprarenal gland, which is just the other name for the adrenal glands. So the suprarenal glands, they are divided into medulla and cortex. So the medulla is mainly producing a lot of catecholamines. So the medulla of the adrenal glands, they are producing catecholamines, epinephrine or epinephrine, and also dopa. Then the cortex is producing a lot of steroid hormones. So the cortex is also divided into zona glomerulosa, zona reticularis, and zona glomerulosa. We have zona glomerulosa, we have zona reticularis. So there are three zones there that are producing the hormones. So we'll appreciate more those zones. So we have those that are producing cortisol, those that are producing corticosterone, aldosterone, and androgens. So even the zona fasciculata, so there are three: zona glomerulosa, zona fasciculata, and zona reticularis. So they are producing steroid hormones like cortisol, corticosterone, aldosterone, and androgens, which are male sex hormones. So androgens are male sex hormones. So be it a man or a woman, male or female, they do produce some amount of androgens, because in the adrenal glands there is a portion that produces androgens. Then we have pineal glands that are producing melatonin. Melatonin is very important when it comes to sleep and awake. For a person to be sleeping or to be awake, this hormone will have a function in it. So we are going to discuss how it's going to influence the cycle, the circadian rhythm. So the normal circadian rhythm is affected by the melatonin. Then the parathyroid glands, they are mainly producing parathyroid hormone that will have an effect on the concentration of calcium. So if the calcium levels are reducing, the parathyroid gland, they can produce a lot of parathyroid hormone that will cause bone resorption. So it will mobilize calcium from the bone, then to also influence absorption of calcium from the GIT, and also minimize excretion of calcium by the kidneys. So it can also enhance reabsorption of calcium from the kidney nephron. So that's the parathyroid hormone. Then the heart can also produce the atrionatriuretic peptide, ANP. So it's some kind of a hormone that will have an effect on other cells of the kidneys and also other cells of the heart itself. Then the kidney can produce hormones like erythropoietin that will have an effect on erythropoiesis, the process by which erythrocytes or red blood cells are produced. So it can go and stimulate the hematopoietic stem cell to differentiate into red blood cells during erythropoiesis. Then it can also produce calcitriol. So it can produce calcitriol and also renin. So these are some of the hormones that are coming from the kidney. Then the digestive tract, of course, there are numerous hormones that are coming from there. So you remember the cholecystokinin, the secretin, the ghrelin hormone, all those are coming from the GIT. So the GIT, the digestive tract, produces a lot of hormones, of which their functions and production, we've also discussed them during GIT, our lectures in GIT. The pancreatic isolates of Langerhans, they are producing insulin and glucagon, and there are also other hormones that are being produced by the pancreas, but the major ones are insulin and glucagon. We'll expand on them once we start looking at the pancreas. Then the gonads, the male and female organs, they are also producing certain hormones. So you have the testes that can, in males, they can produce androgens. So you have testosterone that is being produced, and then you also have inhibin that is being produced by the testes. Then the ovaries, in females, they are producing a lot of estrogens, progestins, and also inhibin. So these are the hormones that are there. So like I said, this is just a summary of what we're going to be discussing in lectures. So we proceed. So what are hormones? How can you define hormones? So hormones, or a hormone, is a chemical messenger transmitted into the blood that is secreted by an endocrine gland. So an endocrine gland is secreting a hormone, then it will be transmitted by the blood. So a chemical messenger produced by endocrine gland, transmitted by blood. That's a hormone. So how do they operate? So in simple terms, hormones, they operate like a radio station. So like a radio or a TV station. So if you have a radio station or a TV station, so those radio, they are more like the cells that are producing the hormone. So the hormone will be broadcasted, to say a signal is broadcasted to the whole body. But only those cells that will have receptors will be able to respond. So that's how the radio works like. We know to say that you can have a radio station, but for you to listen to that station, you need a stereo or a radio. So the stereo is more like the cell. Then it has got something that you can use to tune the radio, to tune to different channels. So as you are tuning, you are introducing a receptor for the signal. The signal is the hormone. So if you don't tune to that signal, to that channel, you are not going to listen to anything. So by you tuning, it means you're introducing a receptor, a specific receptor for that signal. So that's how hormones also operate. The glandular cell is just going to broadcast the hormone. They need to be transported by blood, or the target cells that will have a specific receptor, they will be able to respond. Why? It's because the hormone now will be able to go and bind to the receptor and trigger a cascade of reactions within those target cells. Then delivery time of chemical messengers is less than a minute. The entire blood supply circulates about once every minute. So to say that the transmission of these chemicals will be less than a minute. Why? It's because the cardiac output is 5 liters per minute. Then on average, a person will have 5 liters of blood. So it means that in every other 1 minute, the blood goes into circulation. So if the blood is going into circulation after 1 minute, it means that even the transmission of these hormones, once they are released into the blood, it will be less than a minute, and then it gets to the target cells. That's what it means. So hormones are broken down rapidly, but they set in motion effects that may persist after the hormones are gone, stimulate metabolism, turn on genes, etc. So it means that once the hormones have been secreted, they will go and have an effect on the cells. So even when the hormones are destroyed, their effect can continue. Why? It's because some of these hormones, they will stimulate the genes. So the gene transcription can continue even after the hormone has been destroyed. So some of the effects of these hormones, you can see them maybe after 2 days or 3 days. Why? It's because it will involve the activation of certain genes that will involve transcription, protein synthesis, then you have the effect. But other hormones, the response is immediately. Maybe it can cause contraction of certain cells like muscle cells, contraction of muscle cells. So some of the response is immediately. So you find that once it has been produced and released, then to have an effect on other cells. If these are catecholamines, of course, they can cause vasoconstriction because they can stimulate the smooth muscle contraction. So if the smooth muscle cells lining the blood vessels are stimulated by catecholamines, then they are going to contract. So the effect there could be immediate, but other cells will take a bit of time for them to respond, especially when you're looking at the function of steroid hormones that will involve the receptors that are located inside the cells, in the cytoplasm or in the nucleus. So much of the endocrine system is controlled by the nervous system. The hypothalamus and the pituitary control a wide range of hormones through the trophic hormones. So we have the trophic hormones. These trophic hormones are the ones that are controlling the other hormones. So they are produced by the hypothalamus. So there are two hormones that are being produced by the hypothalamus. We have the releasing hormones that will stimulate the pituitary gland to release other hormones, or we can have inhibitory hormones that will inhibit the pituitary gland to release other hormones. So we have the control of hormones that are being produced. Then the hormones that are coming from the anterior pituitary gland, they can stimulate other hormones. Like the thyroid stimulating hormone is produced by the anterior pituitary gland. Then after its production, it will go and stimulate the thyroid gland to release a lot of T3, T4, tetraiodothyronine and triiodothyronine. So this is the control of hormone production. that is being controlled by the hypothalamus via the function of the pituitary gland, especially the anterior pituitary gland. Because in the posterior pituitary gland, there is production of ADH and oxytocin, antidiuretic hormone and oxytocin. So the two hormones are secreted into the blood by the endocrine glands. An example given there, we have thyroxine, epinephrine, which is also referred to as adrenaline hormone, testosterone, the estradiol, and also insulin. So these are examples of two hormones. So the two hormones, they need to be produced by endocrine glands for them to qualify to be true hormones. Otherwise, we also have neurohormones that are being secreted by nerve cells. An example, antidiuretic hormone and oxytocin. So these are examples of neurohormones. They are being secreted by a nerve cell or the neurons. The pheromones are airborne signals secreted by exocrine glands and stimulate target cells in another animal, usually of the opposite sex. So an example that I can give here are the pheromones that are produced by insects, mammals, and reptiles, and also other mammals that will mark their territory with urine, feces, and scent glands, do so through the action of pheromones. So mammals that will mark their territory, like the lions, all those animals that can mark their territory, they do so by producing pheromones in the urine or the fecal material, such that other animals, as they are approaching that territory, they will know by the scent of the pheromones to say there is another animal that is marking this territory. So all the territorial animals, they are able to produce pheromones that can have an effect on other animals. So they will be able to tell to say this is a no-go zone because this is a property for someone. So if you are crossing there, you are trespassing, then there could be a fight there. So you find that all those animals that are producing pheromones, they can mark their territory. And we also have other pheromones that I can give an example in pigs. In pigs, we have the male pigs that can produce androstenone. So the androstenone are male pheromones that are produced by the male pigs. Then they can have an effect on the female pigs. So the female pigs, for them to come on heat, they need to sense the presence of the androstenone. So the androstenone will stimulate the female pigs to come on heat. Otherwise, if there is absence of the androstenone, those female pigs will never come on heat because they will know to say that what's the reason for them to come on heat if there is no male around them? Because even if they come on heat, there will be no male pigs to mount them or to have sex with them, in other ways, in simpler terms. So you find that there are male pigs that are producing androstenone, and then it will have an effect on female pigs. When you go to a pig farmer, they are going to separate the male pigs from the female pigs. They are going to put them in pens. In those pens, they're just going to use maybe a wire to separate the male from the female so that air can move from the male pigs to the female pigs. That is very key with regards to production in a piggery, because the female pigs, they can only come on heat if they are sensing the presence of the pheromones that are being produced by the male pigs. So those pheromones here, they are called the androstenone. The androstenone. Then we also have interferons. Interferons, these are released by the white blood cells in response to viral infection. So you saw that we have the macrophages and other white blood cells that can produce interferons, and those interferons will stimulate other cells to become resistant to the viral infection. So maybe there's a virus that is invading body cells or host cells, then those host cells that are infected, they'll produce certain chemicals, and those chemicals will stimulate the white blood cells to start producing interferons, and the interferons will stimulate other cells now to develop immunity and also to become resistant to the infection. So interferons are proteins released by cells in response to viral infection. So they stimulate the release of antiviral proteins or enzymes that act to destroy the virus itself. So the other cells will start producing antiviral proteins that can destroy the virus, and interferons also act. They will activate the immune system to attack cancer cells. So other immune systems can be activated by interferons so that you start now attack even cancer cells. And the cytotoxic T cells will be activated, then they'll start attacking the cancer cells or to destroy cancer cells, which is part of immunity. Then we have prostaglandins. The prostaglandins are fat acid based chemicals. So they are fat acid based chemicals that are made in most of the tissue of the body. So we have prostaglandins that are being produced by certain cells. So they'll have a diverse range of actions, including inflammation, fever, and development of pain. So we have cells that are producing prostaglandins, and that prostaglandin can go and bind to pain receptors, and then they can initiate pain sensation, especially in females. When they are having their menses, there are cells that are producing prostaglandins, and those prostaglandins can increase body temperature in females, and at the same time they can go and bind to pain receptors. Then they have muscle cramp. They will tell you to say they need to buy Buscopan, they need to buy Brufen, they need to buy analgesic drugs so that at least they take care of the pain. So that is so because of the prostaglandins. So these prostaglandins, they are binding to receptors. So you find that there are certain females that will produce less of prostaglandins, and some of them, they don't necessarily bind to pain receptors. So you find that certain females, when they are having menses, they will experience pain. Others, when they are experiencing menses, or when they are having menses, they don't experience pain. So there are variations from one individual to another individual because of the different arrangement of prostaglandins and the receptors that are found there. Okay, so many of their actions are associated with control of smooth muscle cells in blood vessels. So control of smooth muscle cells. They can cause vasoconstriction, vasodilatation, depending on the receptors and the different types of prostaglandins. So you have prostaglandins E2, E1, E3, different types, prostaglandins I1. So there are different types of prostaglandins that will have different effects. Okay. So hormones are grouped into three classes based on their structure. So based on the structure of the hormones, they are classified into three classes based on the structure, the anatomical structure or the physical structure of the hormones. So the structure of the hormone is affected by the precursor for the hormones or the raw materials that are being used to synthesize or to produce the hormones. So there are those that are produced that in terms of nature, they are amino acids, or they could be proteins or peptides. Some of them, they are derivative of amino acids. Some of them are derivative of cholesterol, which are called steroid hormones. So according to the structure of the hormones, they are classified into three groups. So the first group, they are called peptides or proteins. So we have peptides or proteins. Then we have the amines. So the amines are derivative of amino acids, especially two amino acids, tyrosine and tryptophan. So the derivative of tyrosine and tryptophan, they are called amines. Then we have steroids. Steroids are derivative of cholesterol. So they are formed from a special type of fat, which is called cholesterol. So they are fat soluble because they are cholesterol in nature. So according to the structure, they are classified into three: peptides, amines, and steroid hormones. Examples of peptides, we have a lot of hormones that are classified as peptides or proteins. So most of the hormones that we'll be discussing, which are water soluble, they are peptides or proteins in nature. Then we have amines. Amines are derivative of tyrosine and tryptophan. So derivative of tyrosine, we have thyroid hormones, triiodothyronine, and thyroxine. Thyroxine is also called tetraiodothyronine or T4. Triiodothyronine is T3. Then we have adrenomedulla, which are catecholamines like epinephrine or adrenaline. This is also a derivative of tyrosine. Then we have a derivative of tryptophan, the hormone that is being produced by the pineal gland, which is called melatonin. So melatonin is a derivative of tryptophan. So if you're lacking these amino acids, it means that you'll be also lacking these hormones. So it will bring about a disorder due to deficiency of these amino acids, because for you to produce these hormones, you need the two amino acids, the tyrosine and the tryptophan. Then we have steroid hormones. Steroid hormones are derivative of cholesterol. So in terms of structure, they look like cholesterol. So we have the hormones that are produced by the adrenal cortex, the steroid hormones that are produced by the adrenal cortex, like aldosterone and cortisol. So aldosterone is being produced by the zona glomerulosa. The zona glomerulosa produces a lot of cortisol. And also the zona fasciculata produces cortisol. Aldosterone is produced by the zona glomerulosa. Cortisol is produced by the zona fasciculata. Then of course we have zona reticularis that produces androgens. Then we have the gonads. The gonads, these are male and female sex organs that will produce certain hormones, like the testes, they'll produce a lot of testosterone. Then the ovaries, they produce estrogens and progesterones and other hormones. Okay, so these are examples of steroids. So they are fat soluble. The amines, some of the amines are fat soluble, others are water soluble, but the peptides mainly, they are water soluble. So most of the hormones are water soluble. So if they are water soluble, in terms of triggering a response to the cells, they are attached to receptors that are embedded within the plasma membrane because they can't penetrate the cells. But the steroid hormones, because they are fat in nature, they are able to penetrate the hormones to enter they are able to penetrate the cells to enter the cells via the plasma membrane because they are fat soluble. But for them to be transported, they need to bind to plasma proteins because they are fat soluble. They don't dissolve easily in water. So in the water, they can't dissolve. So the only way they can be transported is by binding to plasma proteins. Then once they are released now to the target cells, they will be able to penetrate the target cells, and then they will be transported by intracellular proteins to the target receptors. Then they can attach to the receptors to trigger chemical reactions or a cascade of reactions that will bring about cell response. So the chemical structure and the synthesis or production of hormones. So what is the chemical structure and the production of hormones? So this we are also going to discuss more in biochemistry. So we're not going to waste much time here to look at the chemical structure and the production of these hormones, of which we've already done in biochemistry. So there are three general classes of hormones. So the first class are called proteins and peptides. So the proteins and peptides, including hormones secreted by the anterior and posterior pituitary gland. So those are proteins and peptides in nature. So they can easily dissolve in plasma for transportation. They don't need to bind to plasma proteins. Then you have hormones that are produced by the pancreas, like insulin and glucagon. They are also protein and peptides in nature. The parathyroid glands, the parathyroid hormone, they are producing parathyroid hormone, which is also water soluble. So it's protein or peptide in nature. So these are classified as proteins and peptides. They don't need to bind to plasma protein for their transmission or for their transportation. Steroid hormones are secreted by the adrenal cortex. So the adrenal cortex is producing a lot of cortisol and aldosterone. The ovaries, they are producing estrogens and progesterones. The testes, they are producing testosterone, and also the placenta can produce estrogens and progesterones. So these are fat soluble. So they are called steroid hormones because they are derived from cholesterol. So for them to be transported, most of them, they need to bind to plasma proteins, and their receptors, they are found inside of the cells, be it in the cytoplasm or in the nucleus. So these are steroid hormones. So they are able to penetrate the phospholipid bilayer because they are fat soluble. Then they will enter the cells to go and bind to their specific receptors. So the target receptors are inside of the cells. So intracellular receptors for steroid hormones. That's why the steroid hormones are being abused by these guys who go to the gym. So to say they are lifting a lot of metals, they are working out. So they are going to be taking a lot of steroid hormones like testosterone or progesterones, especially testosterone. So if they are taking a lot of testosterone, to say testosterone will be able to be transported to the muscle cells. It will cross a phospholipid bilayer of the muscle cells to enter the muscle cells. Then it will go and bind to receptors within the cells and also in the nucleus. It will trigger transcription. Then you have a lot of messenger RNA that will go to the rough endoplasmic reticulum to be translated into a protein for the protein synthesis, and those proteins now, they'll become proteins in a muscle cell. So the muscle cell now, it will accumulate a lot of proteins, and these are structural proteins and also contractile proteins like actin, myosin. So you have more of those proteins, and then the cell will be increasing in size. It will bring about hypertrophy of the muscles. That's why if you are working out and then you want your muscles to grow very fast, they will be abusing a lot of steroid hormones, okay, because of the mode of action for steroid hormones. You understand as we are proceeding with our lectures. So the derivatives of the amino acids, tyrosine, so we say that the hormones that are produced by the thyroid gland, they are producing thyroxine and triiodothyronine. So these are derivative of tyrosine, and also the adrenal medulla that is producing a lot of catecholamines, epinephrine and norepinephrine. So these are catecholamines that are produced by the adrenal medulla, and of course you have those that are being produced by the pineal gland. The melatonin is a derivative of tryptophan, which is not here. So there are no known polysaccharides or nucleic acid hormones. So there are no hormones that are carbohydrates in nature, or DNA or RNA in nature. So there are no such hormones. Okay, so most of the hormones, it could be proteins, peptides, steroid hormones, or derivative of amino acids. So these are the that we know so far. And this, if you want to discover your own. Okay, so adding more information to proteins and polypeptide hormones. So the polypeptide hormones or protein hormones are stored in secretory vesicles until needed. So they are produced and stored within secretory vesicles. When they are needed, they will be released. And most of the hormones in the body are actually peptide hormones or proteins. So most of the hormones are peptide in nature or proteins. So those hormones range in size from small peptides with as few as 3 amino acids, like thyrotropin releasing hormone, so it has less than 3 amino acids, to proteins with almost 200 amino acids, like growth hormone or prolactin. So these hormones, they could be smaller in structure or larger. So those that are smaller, they are called peptides. Those that are larger, they are called proteins. So any hormone that has got less than 100 amino acids is referred to as peptides or polypeptides. But if it's composed of more than 100 amino acids, then they are referred to as peptides. So the hormones that will have more than 100 amino acids coming together to form polypeptide chains, they are referred to as proteins and not peptides. So the protein and peptide hormones are synthesized on the rough endoplasmic reticulum of different endocrine cells. So you have different endocrine cells. They are being produced in the rough endoplasmic reticulum because that's where you find the ribosomes that are responsible to translate message that is brought in by the messenger RNA for the production of proteins. So here it is. So in the rough endoplasmic reticulum, you have protein synthesis taking place there. They contain ribosomes, 40S, kistase. At the center, you have the transfer RNA that will translate the codon or the message that is contained within the messenger RNA for the production of a particular protein. So these proteins that are being produced in the rough endoplasmic reticulum, they are called pre-prohormone. So they are called pre-prohormone. They are not active as yet. Then they'll be packaged, and within the rough endoplasmic reticulum, there is modification that is taking place. So the pre-prohormone will be converted into prohormone by cleavage of certain amino acids. So there is a bit of modification that is taking place to convert the pre-prohormone into prohormone. Then the prohormone will be converted into the actual active hormones in the Golgi apparatus. So in the Golgi apparatus, you have more modification of these hormones, and they will be now converted into mature hormones, or the actual hormones stored within vesicles, waiting for signal for exocytosis. So once there is a signal for exocytosis, there will be now release of these hormones in form of exocytosis process. So the cell now will release the hormones. So this hormone now will be transported by the circulatory system to the target cells. So these are proteins, polypeptide hormones. So you can see that you have the DNA, there is transcription taking place there. The message that is contained within the DNA will be transcribed into the messenger RNA. Then the messenger RNA will leave the nucleus to go to the raffined plasmic reticulum to interact with the ribosomes. The ribosomes will translate the message to combine amino acids to form the polypeptide chain and packaged within the vesicles. So you know to say that the hormones that are being produced initially here, they are called pre-prohormone. Then there's a bit of modification to convert them into prohormone, stored within the vesicles, transported by the molecular motors in form of vesicles to the Golgi apparatus for the further modification. Then they'll be converted into hormones stored within the vesicles. So you can see a lot of secretory vesicles that are containing the hormones, just waiting for signal for exocytosis. So the signal for exocytosis, it will come as a stimulus. So it will stimulate this cell to release the hormones. So this stimulus can trigger an increase in intracellular calcium and cyclic AMP. You know that the calcium will bring about exocytosis because the calcium can go and bind to V snare, T snare. So T snare, V snare interaction, it will cause exocytosis of the hormone. Then the cyclic AMP, you know to say the CAMP or cyclic AMP, it can go and activate protein kinase. And then the protein kinase, some of them, they are also involved in exocytosis. So this stimulus will stimulate exocytosis of the hormones. Then the hormones will enter the cardiovascular system. Then they will be transported to the target cells for their response. So it's the same process that is taking place here, even the exocytosis. So I've already explained this process. So we make progress. So moving on to steroid hormones. So the steroid hormones are usually synthesized from cholesterol and are not stored. Because we said the proteins, peptides, after production, they are stored within vesicles, just waiting for signal for exocytosis, which is a stimulus that can cause an increase in calcium and also cyclic AMP for them to exocytose the protein hormone or peptide hormones. But the steroid hormones are being produced by certain cells, the glandular cells, and once they are produced, you know to say they are not being stored because they are derivative of cholesterol, so they are being produced from cholesterol. So they are not usually stored. After production, they will be released. So once there is a signal for exocytosis for steroid hormones, then it will bring about exocytosis of these particular hormones. So the chemical structure of steroid hormones is similar to that of cholesterol, and in most instances, they are synthesized from cholesterol itself. So they are lipid soluble and consist of 3 cyclohexyl rings. So they consist of 3 cyclohexyl rings and 1 cyclopentyl ring combined into a single structure, just like the way cholesterol looks like. So these hormones, they look like cholesterol. So the steroids are highly lipid soluble, and once they are synthesized, they will simply diffuse across the cell membrane and enter the interstitial fluid, and then the bloodstream. So they are produced, released into the interstitial spaces. They'll enter the interstitial fluid, then they will also move to the cardiovascular system. So once they are produced by these cells, they are able to cross a phospholipid bilayer because they are lipid soluble. So you don't need a particular channel for it to move. It's lipid soluble. It can just move between the phospholipid bilayer to move from intracellular to extracellular down the concentration gradient. So these are steroid hormones. So the production of steroid hormones is taking place within the endoplasmic reticulum, and some of the reactions they are also taking place within the mitochondria. So the production of steroid hormones. So you can see you have cholesterol here, the adrenal cortex, you have the zona fasciculata, zona glomerulosa, zona reticularis that are producing steroid hormones. So from the zona fasciculata, you can produce cortisol and corticosterone. From the zona glomerulosa, the production of aldosterone. Then from the zona reticularis, you have production of androstenedione. So these are steroid hormones that are being produced. So these pathways, you're going to discuss this in biochemistry. Maybe you've already discussed that in biochemistry. So I'm not going to bother you to look at the structure, the hormones that are involved in the production of steroid hormones. But just not to say from the zona fasciculata, you have the production of cortisol and corticosterone. Then from the zona glomerulosa, production of aldosterone. Then from the zona reticularis, you have the production of androstenedione. That should be enough in physiology. So some of the chemical reactions they are taking place within the mitochondria and others within the smooth endoplasmic reticulum. So in the smooth endoplasmic reticulum, that's where you have production of steroid hormones. Remember, steroid hormones are not proteins. So you don't need ribosomes for the production of steroid hormones. So steroid hormones are being produced within the smooth endoplasmic reticulum, and also some of the reactions are taking place within the mitochondria. But the pathways, you don't need to remember all the pathways that are involved in the production of steroid hormones. The derivatives of amino acids, tyrosine and tryptophan. So we have two groups of hormones that are derived from tyrosine. So we have those that are being produced by the thyroid gland, which are called thyroid hormones, the T3, T4, triiodothyronine, tetraiodothyronine. So they are synthesized and stored in the thyroid gland, incorporated into the macromolecules of the protein thyroglobulin. So the thyroglobulin is the one now that will be releasing the T3, T4 slowly, which is now stored in large follicles within the thyroid gland. So you appreciate this when we start looking at the production of the thyroid glands and the function of the thyroid glands, which are also referred to as metabolic hormones. So they will increase the metabolism of the body. The adrenal medullary hormones, so these are catecholamines that are being produced from the tyrosine hormone, tyrosine amino acids. So the adrenal medullary hormones, epinephrine and norepinephrine, are formed in the adrenal medulla of the adrenal glands. So the adrenal normally secretes about 4 times more epinephrine than norepinephrine. So they'll produce more of epinephrine as compared to norepinephrine. The catecholamines are taken up into preformed vesicles and stored until secreted. So here they are taken up by vesicles, stored, just waiting for signal for exocytosis of catecholamines. So the catecholamines are released from the adrenal medullary cells by exocytosis. So the process is the same because they are stored in vesicles, so they are going to be released by exocytosis. But they are fat soluble, so for them to be transported, they need to bind to plasma proteins. So once catecholamines enter the circulation, some of them they can bind, not necessarily binding to plasma proteins, because most of the catecholamines actually are water soluble, so they don't need to bind to plasma proteins. Okay, so they will be transported as dissolved hormones in plasma. So they can exist in plasma in free form or conjugated with other substances. So the conjugated with other substances are the ones that are binding to plasma proteins, but most of them, they are actually transported in free form as dissolved hormones in plasma. The amines hormones, the structure, so you have the catecholamines, they are derivative of tyrosine, so you can produce these catecholamines. So you can see you have dopamine, norepinephrine, and epinephrine. The structure is similar. There's just some enzymes that are changing a bit of the functional group, the hydroxyl group, and other groups that are changing to convert them from one form of catecholamine into another form. So these are the three catecholamines, the major ones: dopamine, norepinephrine, and epinephrine. In terms of structure, they are similar. Then we have the thyroid hormones, the tetraiodothyronine and triiodothyronine. So we have the tetraiodothyronine with 1, 2, 3, 4 iodine attached to the molecule, hence the name tetraiodothyronine. Then we have the triiodothyronine with 1, 2, 3 iodine that is attached there. Okay, so these are derivative of tyrosine amino acid. The production of catecholamines. So again, here you don't necessarily need to remember all these enzymes, but just at least understand the different types of catecholamines. So the catecholamines, they are being produced from tyrosine. So first, the tyrosine is converted into the phenylalanine. So you can see the tyrosine is being converted into phenylalanine. So the phenylalanine, we have this phenylalanine, will be worked on by this enzyme, the phenylalanine hydroxylase. So the hydroxylase is going to add the hydroxyl group to the phenylalanine. So you can see this is the structure of the phenylalanine. Then if you add the hydroxyl group on this carbon, or this structure, the ring structure, then it will be converted to tyrosine. So you can see here the conversion of phenylalanine to tyrosine. Then the tyrosine can also be converted to tyrosine. So we have another enzyme that can remove the hydroxyl group to convert it to phenylalanine. Okay, so now this tyrosine will be worked on by another enzyme, which is called the tyrosine hydroxylase. So we have the tyrosine hydroxylase that will add another hydroxyl group. So you can see another hydroxyl group that is being added here. So it will be converted from tyrosine to dopa. Dopa stands for dihydroxyl phenylalanine, meaning that you've only added two hydroxyl groups. Hence, di means two. Hydroxyl, these are hydroxyl groups that are being added to phenylalanine. So dopa stands for dihydroxyl phenylalanine, because we've now added two hydroxyl groups by these hydroxylase enzyme. Then when you have this dopa, there is another enzyme, aromatic L-amino acid decarboxylase. So the decarboxylase is going to remove the carbon dioxide from the carboxyl group here. So once you remove the carbon dioxide from the carboxyl group here, meaning that you are converting it now from an acid into other chemicals, so the dopa will be converted into dopamine. Once you have a decarboxylase that is removing the carbon dioxide, from the carboxyl from the dopa, then it will be converted into dopamine. So you can see now you don't have the carboxyl group here. So it has been converted into dopamine. Then if you have dopamine beta hydroxylase, so it's going to add this hydroxyl group, then it will be converted from dopamine into norepinephrine. Then norepinephrine, you have phenyl, ethanoalanine, N-methyltransferase. So this is the N-transferase. It's just going to transfer the methyl group. So you can see it's going to transfer and add the methyl group here. So once it will transfer this methyl group, then it will be converted into epinephrine. So from norepinephrine to epinephrine. So this is the production of catecholamines. It's more of biochemistry, but in physiology you just need to know the different types of catecholamines, how they'll come about, and the function of these catecholamines. That should be enough. The physiological function. So the synthesis or the production of catecholamines, we've already discussed that in the previous slide. So we make progress. The production of melatonin. So melatonin is a hormone that is being produced from tryptophan. So it's a derivative of tryptophan. So you can see the L-tryptophan amino acids that can be converted into melatonin. So we have also a series of enzymes. So you have this tryptophan hydroxylase. So it's going to add the hydroxyl group to convert it to L5-hydroxyl tryptophan. Then we have another enzyme, a decarboxylase, that will remove a carbon dioxide from this group here. Then it will be converted into serotonin. So the serotonin 5-hydroxyltryptamine. So the 5-hydroxyltryptamine is another name for serotonin. Okay, so in other textbooks you will see 5-HT. That simply stands for 5-hydroxyltryptamine, which is the other name for serotonin. Then you have an enzyme. So this is the N-acetylase, meaning that it will just add another group, the acetyl group, then to be converted into N-acetyl-5-serotonin. Then you have another enzyme, the transferase, the methyltransferase, that is going to transfer the methyl group and then to be converted into melatonin. So this is more of biochemistry, like I said, but just need to know that melatonin is being produced from tryptophan. You have a series of reactions that is taking place. Okay, so the pathway, then it's biochemistry, but for now physiology, you just need to know that serotonin can also be converted into melatonin, but these two neurotransmitters or neurohormones, they are produced from the tryptophan amino acid. The mechanism of hormone release. So what is the mechanism of hormone release? So there are three mechanisms by which hormones are released. So the first one is humoral mechanism. Humoral in response to changing levels of ions and nutrients in the blood. So depending on the concentration of ions and nutrients in the blood, certain hormones, they can be released. You agree with me to say parathyroid hormone is going to be released when you have hypocalcemia. So in hypocalcemia, if there is low concentration of calcium, then parathyroid hormone is going to be released. So it's responding to the level of ions in the blood. Insulin glucagon is interested in the concentration of glucose in the blood. So in case of hypoglycemia, there will be release of glucagon. Hyperglycemia, when you have an increase in glucose levels, you have a release of insulin. So they are responding to the concentration of glucose or to the concentration of nutrients in the blood. So that's one mechanism. Humoral, meaning that it's responding to the concentration of ions or nutrients in the fluids, hence the name humoral mechanism of hormone release. Calcitonin, if you have more calcium, calcitonin will be secreted. If you have less calcium, parathyroid hormone will be released. So that is called humoral mechanism. Then we also have neuromechanism. The neuromechanism of hormone release is whereby the nerve cell is stimulating the glandular cell to release some hormones. So it's called neuromechanism. So the neuro, you have the nerves that are innervating the glandular cells, especially the adrenal medulla is innervated by nerve cells. So you have the sympathetic nerve cell that is innervating the adrenal medulla. So once you have sympathetic stimulation, it will stimulate the adrenal medulla to release a lot of catecholamines. Epinephrine and also norepinephrine will be released. So it's because it's the nerve cell that is stimulating the glandular cell to release other hormones. That mechanism is called neuromechanism. So neuromechanism of hormone release. And then the last one is hormonal, whereby a hormone is stimulating the release of another hormone. So that is called hormonal. So stimulation received from other hormones. So I told you to say that the pituitary gland, especially the anterior pituitary gland, they are producing a lot of stimulatory hormones that can stimulate other glandular cells to produce other hormones. The thyroid stimulating hormone that is being produced by the anterior pituitary gland is going to stimulate the release of the thyroid hormones from the thyroid gland. So that is an example of hormonal. So the hypothalamus is producing thyroid releasing hormone, then the thyroid gland, the pituitary gland is producing thyroid stimulating hormone, then the thyroid gland will produce thyroid hormones like thyroxine, triiodothyronine or tetraiodothyronine. So one hormone is stimulating the release of another hormone. So it's called hormonal mechanism by which the hormones are being released. So to expand on that knowledge, there is a picture that is explaining the same here. So we have the humoral, whereby the hormone that is being released is responding to changing levels of ions or nutrients in the blood, like the parathyroid hormone. So if you have low levels of calcium, hypocalcemia, the parathyroid glands will be stimulated to release parathyroid hormone. That will now try to increase the concentration of calcium by causing bone resorption. So it will go to the bone and cause mobilization of calcium from the bone deposits. Then it will go to the GIT to increase absorption of calcium from the GIT. It will go to the kidneys to cause reabsorption of calcium from the GIT. So that's the parathyroid hormone. It's responding to the levels of ions, which is calcium, in the blood. Then we have neural mechanism. You can see we have this sympathetic nerve that is innervating the adrenal medulla to stimulate the glandular cells here that are producing catecholamines. Then the catecholamines will be secreted into the interstitium, transported by blood, then to have an effect on other cells and the smooth muscle cells of the blood vessels. Then the last one is hormonal mechanism, whereby the hypothalamus is releasing hormones to cause the anterior gland to release a lot of hormones like the thyroid stimulating hormone, adrenocorticotropin hormone, and also follicle stimulating hormone or luteinizing hormone. So those will go and affect other glandular cells to release other hormones. So the thyroid stimulating hormone is stimulating the thyroid gland to release thyroid hormones. Then the adrenocorticotropin hormone is stimulating the adrenal cortex to release steroid hormones. Then we have the follicle stimulating hormone, luteinizing hormone that can go to the gonads, like the testes and the ovaries, to stimulate the production of testosterone, production of progesterone, and estrogens. So this is hormonal mechanism. So the endocrine system uses the cycles, negative feedback, and antagonistic pairs of hormones to regulate physiological functions. So negative feedback is going to regulate the secretion of almost every hormone in the body. So the negative feedback mechanisms are common as opposed to positive feedback mechanisms. The cycles of secretions maintain physiological and homeostatic control. So you have cycles in which certain hormones are going to be secreted. So these cycles can range from hours to months in duration. So other cycles is just within hours, others can take months. Due to seasons, they are going to be secreted. So there are seasons that can affect secretion of particular hormones. So this is how the hormones are released from the body. So the most common are negative feedback mechanism. So they are most common. So endocrine glands, they oversecrete hormones because of the stimulatory factors that are there. So those hormones that are being secreted, they can function as a negative feedback to inhibit the pathway that is producing that particular hormone. So the product that is going to be there is going to inhibit the pathway producing that particular hormone. So this is a negative feedback mechanism. I'll give you an example. So an example given here, the previous slide, I told you to say that we have the thyroid stimulating hormone that is being produced by the anterior pituitary gland that will go to the thyroid gland to stimulate the production of thyroid hormones. So if the concentration of thyroid hormones are increasing, they can inhibit the release of thyroid stimulating hormone. So if the production of thyroid stimulating hormone is being reduced or being inhibited, then the release of thyroid hormones will also reduce. So that's a negative feedback mechanism. The product will inhibit the pathway producing the hormone. So that's a negative feedback mechanism. But the positive feedback mechanism is the opposite. The product here is going to enhance the pathway producing that hormone. An example I can give you is the pathway for luteinizing hormone. You know, to say estrogens, they are supposed to inhibit the production of luteinizing hormone. But towards ovulation, the more estrogen that is being produced by the ovaries, they are going to overstimulate the luteinizing hormone. That will bring about ovulation. So that becomes a positive feedback mechanism, because now the higher concentration of estrogens is not inhibiting the production of luteinizing hormone, is overstimulating the production of luteinizing hormone. That will bring about ovulation. So that's an example of positive feedback mechanism. But we have very few positive feedback mechanisms in the body as compared to the negative feedback mechanism. So most of the hormones, they are being controlled by negative feedback mechanisms. So regulation of hormone secretion, then we have the neuromechanism and the feedback control mechanism. The neuromechanism will involve the secretion of catecholamines by the adrenal medulla, which I've already explained. The feedback mechanism, you can either have the positive feedback mechanism or a negative mechanism, which I've also explained. So this is the last part of our lecture. This is the introductory lecture, part 1. We're going to look at measurement of hormones concentration in the blood. How are these hormones measured? Because now you know that the hormones are being secreted, then they are transported to the target organs. They will have an effect on target organs. But how can you quantify these hormones? So most hormones are present in the blood in extremely minute quantities. So they are extremely in minute quantities, meaning that they are in smaller quantities. So some concentrations are as low as 1 billionth of milligram. So it means that the concentrations are extremely low. So there are techniques that are employed to quantify the hormones. So therefore, it is very difficult to measure these concentrations by usual chemical means. So other usual chemical means, it's very difficult for us to quantify these hormones in circulation or in our samples. It could be blood sample, urine sample, plasma sample, or body fluid samples. So it's very difficult for you to quantify using just normal chemical means of measuring chemicals of the body. So an extreme sensitive method, however, was developed about 40 years ago that revolutionized the measurement of hormones, their precursors, and their metabolic end products. So there are techniques, there are 2 techniques that you can use to quantify hormones, of which we are also going to discuss in microbiology in details. So in physiology, we're going to mention and how these mechanisms work, or these techniques work. So the measurements of hormone concentration in the blood, the 2 methods that we can use is the radioimmunoassay and enzyme-linked immunosorbent assay, which is also referred to as ELISA. So I'm going to explain how it's going to work. So when you remove this presentation on slideshow, underneath there are notes. You can read extra information underneath there. I always add notes, especially where there are pictures or where there are abbreviations that you can't understand. So I always add extra amount of notes for students to understand. So how does the radioimmunoassay work? So this is, you are going to use competition to quantify the hormones. So binding competition. So you need a radioisotope, which is more like an antigen specific for antibody. Remember, antibody are proteins that are produced by immune cells that are specific for a particular antigen. So once they recognize the antigen, they'll go and bind and form antigen antibody complex. So the interaction between the antigen and the antibody, we are going to take advantage of that interaction to quantify a hormone. How does it work? So we have the radioisotope antigen that we know the concentration of this radioisotope that will react with the antibody. Then the same antibody can also react with the hormone that you want to quantify. So the concentration of the hormone you don't know. So now we are going to introduce an assay where you have minimum amount of the antibodies that can react with the radioisotope antigen, and the hormone will also contain a particular antigen that can interact with the antibody. So here is the issue of competition. So if you realize to say that after interaction, after you mix in solution, and all the radioisotope is binding with all the antibodies, then you will know to say that you don't have this hormone that you want to quantify. If all the 100% of the radioisotope are binding to the antibody, then you know to say that this radioisotope didn't have a competitor to bind with the antibodies. So you know to say that the concentration of the hormone is almost zero. But if you realize to say that the radioisotope antigen, 50% of it is reacting with the antibody or is binding with the antibody, then you know to say that the other 50% is actually the hormone that is binding to it. So they are competing. So if 50% is binding, you know to say that the other 50% is bound to the hormone that you want to quantify. Then you'll be able to come up with the ratio or the concentration of the hormone. That's how the radio immunoassay work. Then we have enzyme-linked immunosorbent assay, ELISA. ELISA, I'm going to explain using the diagram, this diagram here. How does it work? So you have a plastic well that contains 96 of these wells. So the wells, they are more like pits in this plastic plate. You are supposed to coat the plate with the first antibody. So the antibody, the first antibody, is specific for the hormone that you want to quantify. So the antibody, of course, they will bind to the antigen. So it means on this hormone, it has got an antigen that can interact with the first antibody. So after coating the plate with the antibody, the first antibody, you are supposed to introduce your sample that you are suspecting to have the hormone that you want to quantify. Then after introducing your sample, you need to wash the plate. So if the hormone is there, it's going to form an antibody antigen complex. Even if you wash, it won't be washed away. It will still be there. Then you introduce a second antibody that can bind to a different site on the hormone. So the same hormone has got another antigen with a different binding site for antibody 2. So the antibody 2 is going to bind. You wash again. If this hormone was there, then the antibody 2 won't be washed away. It will still be there. So this complex will still be there. Then you introduce your antibody 3. The antibody 3 has got a binding site for antibody 2. So it will go and bind to the antibody 2. And this antibody 3 is linked to an enzyme. So if the hormone is there, the antibody A will bind to the hormone. Then the antibody 2 will come and bind to a different site on this hormone. Then the antibody 3 will come and bind to antibody 2. It's specific for antibody 2. So even if you wash it away, this complex will still be there. So the antibody 3 has got, it's linked to an enzyme. This enzyme will now be able to work on the substrate to convert them into a product. So the product will give you a particular color. So the color intensity is proportional to the amount of the hormones or to the amount of the complexes that are formed there. So you're going to use fluorescent mechanism. Okay, so in biochemistry, there is a spectrometer that you can use to measure or to quantify the intensity of the product, the color intensity of the product. So by checking the color intensity of the product, you are able to quantify the concentration of the hormones. So this is ELISA. This is how it works. So you're going to quantify. But if you didn't have this hormone, it means during washing, this complex won't be there. So the substrate won't be converted to a product. You're not going to get a specific color, and the intensity, the color intensity won't be there. So you know to say that in this sample that you have, you don't have this hormone. But if the complexes are more, even the products will be more, and the color intensity will be more. So you'll be able to quantify the product. So use the spectrophotometer to come up with a color intensity. So we have the fluorescent product P that will give you the color intensity. Then using those techniques or machines, you'll be able to quantify the hormone, the concentration of the hormone. So it's not just the hormone, even the viruses, you can use this technique to quantify the viruses if they are there or not. Why? It's because you just need antibodies that are specific for a particular antigen that is associated with these viruses. So you can quantify even coronavirus, you can quantify different types of viruses because we have different types of reagents. The reagents will contain the antibodies that we are going to use now to form this complex. Okay, so you discuss more in microbiology and also in pathology, but this is how you can quantify the hormones. You'll be able to know that the hormones are there. So this is all for the introductory lecture. Thank you very much for your attention. We end here for today.